Good day, everyone, and welcome. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Biomodels LLC, I'd like to welcome you to Immunological Methods, Understanding and Enhancement of Preclinical Animal Models of Disease. My name is Elizabeth Lamb, and I'm the host and the moderator for today's event. At this time, I'd like to introduce our presenter for today. He is Dr. Dominic R. Beal, PhD, scientist with Biomodels LLC. Welcome, Dr. Beal. The presenter ball is yours.
All right, thanks for the introduction. Okay, so I'm going to talk about these immunological methods and how they can enhance preclinical animal models of disease. A bit of background about Biomodels, the company. We are a preclinical contract research organization located in Watertown, which is just outside of Boston, Massachusetts. We were founded in 1997 out of the Brigham and Women's Hospital, and we specialize in highly translational models of human diseases, and we work with a wide variety of clients, transitioning drugs and biologics from concept into patients ultimately. So far, we have helped facilitate 44 compounds to get into patients, and some of our therapeutic expertise is in the fields of oncology, inflammation and autoimmunity, cardiovascular disease, metabolism disorders, neurology and psychiatry disorders, pulmonary disease, fibrosis, and cancer supportive care. Here's a little outline of the talk I'm going to give today. First of all, I'm going to do an introduction and focus on what is immunology, flow cytometry, and cell sorting. And then I'm going to talk about three of the models that we run here, GVHD, EAE, and IBD. And then I'm just going to do a summary and some acknowledgements at the end. So, immunology. It's obviously the study of the immune system, and basically that means the immune cell types, cell-cell interactions, how they differentiate and mature, how they get to where they're supposed to be getting to in the body, and what are their effective functions in both healthy and diseased states. I found this little picture on just a Google search. And it's not a great picture, but it sums up a lot of the complexity of the immune system, but it also highlights a lot of the misunderstandings. So first of all, there's this division between innate and adaptive, and the innate's labeled a rapid response and the adaptive is labeled a slow response, which isn't strictly true because cells like macrophages and neutrophils and eosinophils are all implicated in a lot of chronic conditions. And then, of course, when you've got memory B cells and memory T cells, those can act very rapidly, and that's basically the whole point of forming memories for that rapid antigen-specific action. What's nice about this is it does highlight this overlap between the adaptive and innate immune responses. Obviously, that's not just limited to T cells and natural killer T cells. There's a lot of cells that overlap the two systems. So now what I want to do is just focus in, just to highlight the complexity of the immune system. I want to focus in on these CD4 T cells. Obviously, that's just one of many, many different types of cell. So this picture is from a 2002 Nature review. This is back when I was in undergrad learning about immunology, and it was super fun and easy because you've got a TH cell. You get the laser on the back. You've got a TH cell here, another TH cell, and you give it IL-12, it becomes a TH1 cell, starts producing interferon gamma, or you can give it IL-4, it becomes a TH2 cell, starts producing more IL-4, and they do two distinct and non-overlapping functions. So that was 2002, and then now the situation in 2016 is this mess. This is, again, just looking at those CD4 T cells, the T helper cells. There's 5 different subsets that this one diagram is talking about, and within the Treg subset, there's multiple subsets as well. But basically, the naive T cell gets signals, cytokine signals and cell signaling molecule signals. They go into the cell, upregulate transcription factors, signaling molecules, and that results in differentiation into one of these subsets and production of specific effector molecules and cytokines. And each one of these subsets has a distinct role. So Th1, they're involved in activating macrophages and defending against intracellular pathogens. Th2, eosinophils, B-cell class switching, defense against extracellular parasites such as helminths, and so on. That's the situation that we're dealing with in 2016 with just the T helper cells. So it's a very complex system. Another way to look at this is a naive cell in the middle here, and depending on the cytokines that you expose it to, it can then differentiate into all these different subsets. So you give this cell a little bit of IL-12, interferon gamma, and you've got a Th1 cell. If you add in a little bit of IL-4, you can get a Th2, Th1 transition cell. A bit more IL-4, it becomes a Th2 cell, and so on. And the upshot of this is that the cytokine profile can be used to evaluate the type of immune response that you're dealing with. So, like I said, Th1 is characterized by IL-12, interferon gamma, etc. Th2, IL-4, 5, and 13. And the reason that's important to know these things is because you can take plasma or tissue, and you can assess the makeup of cytokines and decide what sort of immune response is happening. So we have multiplex ELISA capabilities at Biomodels. We can assess up to 50 cytokines simultaneously in one well of a 96-well plate. So you need a very, very small volume to do that many cytokines, about 25 microliters of plasma, for instance. On that note, I just want to do this little example of why it's important to understand these different cell types and what they're doing. So we do a lot of pulmonary studies here. This data isn't actually generated by Biomodels. This is published data, but this cockroach allergen-induced asthma model is one that we can run. We also run a house dust mite-induced model and obviously the ovalbumin model. One of the main readouts in these models is looking at the cells that are coming into the bronchoalveolar lavage. And so here we have a slide of a mouse exposed to HBSS, that's just the vehicle control, and then a mouse exposed to cockroach allergen. And the real difference is the slide on the left is basically 100% macrophages, and the slide on the right, there's a lot of lymphocytes in there, there's a little black dots, and then eosinophils, these guys with the pink cytoplasm. So there's definitely a difference there, different cell infiltrate. If you quantify that, you can see that lymphocytes, neutrophils, and eosinophils significantly increased after exposure to cockroach allergen, but macrophages aren't doing anything whatsoever. So what's the point in looking at macrophages? So first of all, you have to know that macrophages are an important antigen-presenting cell, especially in the lungs. So antigen presentation is basically how T cells become activated. So the antigen-presenting cell here, the APC, is able to deliver two signals to the T cell. One of them is the peptide MHC TCR complex, and the other one is co-stimulation. If both of those signals are present, the T cell achieves an activation threshold and is able to go on and differentiate and proliferate. If the APC is expressing inhibitory stimulation molecules, then that signal one is sort of counteracted by the inhibitory signal, and you can have different type of activation or anergic or apoptotic T cells. And so the third option would be if the APC is immature or inactivated and not expressing any co-stimulation molecules at all, signal one isn't enough, you again get anergic or apoptotic T cells. So this just demonstrates that the antigen-presenting cell, by virtue of the molecules that it is expressing on the cell surface, is able to determine the style of immune response that the T cell is exhibiting. So then we saw those macrophages weren't changing in number, but maybe we can identify macrophage subsets. There's known to be M1 and M2, which is sort of analogous to Th1 and Th2, then resident versus inflammatory, etc. So if you run some FACS analysis on these macrophages, and you look for these two co-stimulation molecules, CD80 and CD86, those activate T cells, you can see that in mice that are exposed to cockroach allergen, their macrophages have a very much reduced level of expression of CD80 and CD86, and that's indicative of an M2 phenotype, and that's what you'd expect to see with a Th2-mediated response that we get with asthma. So looking at the macrophage function can tell you a lot about the mechanism of action of the T cell maturation and function, and the long-term effects on the quality of the immune response. So that segues nicely into the next section, which is flow cytometry. So I'll just give a very basic overview of this. So you can obtain a single cell suspension, and that can be from just about any organ. These are some of the ones we run commonly here. So you then stain those cells with fluorescent conjugated monoclonal antibodies that are specific to whatever marker is on the cell of interest. So here's a cell. Let's, for example, say that we're looking for antigen-specific Tregs that are producing effector cytokines to act as immunosuppressants. So here's the cell that we're looking at. The first three markers to identify a Th cell will be CD45, CD3, and CD4. And then from there, we can look at intracellular markers, FOXP3, that's a transcription factor that is expressed in Tregs. And then if we wanted to see antigen specificity through the TCR, we could look at the phosphorylated version of ZAP70, which is a signaling molecule attached to the TCR. And then we can look at intracellular cytokines, such as IL-10, which is a Treg effector cytokine inducing immunosuppression. So if our cell was positive for all 6 of these markers, it would be a pretty safe bet that we're looking at an antigen-specific Treg that is able to produce immunosuppressive cytokines. So flow cytometry itself, this is obviously a very, very simplified schematic. Take your single-cell suspension, and it's taken up by the machine, and through a series of extremely well-engineered and complex fluidic systems, that stream of single cells is funneled down into this single laminar flow, and then that passes in front of lasers. So depending on the machine that you're working with, you can have one or more lasers. The laser light is scattered and also is absorbed by fluorescent molecules and then emitted at specific wavelengths. That light is then passed through specific filters and picked up by detectors, and then goes through a whole mess of complicated software to get the images that I'm sure you've all seen. I'll show a few of those in a little bit. So obviously, flow cytometry has a very wide general application across many different disease models, and it's a pretty good technique because it requires only a small number of cells. We usually work with a maximum of about 1 million cells per analysis, and depending on the machine you're using, you can assess between 4 and 20 or more markers simultaneously. To give you some perspective, 1 million cells is about 1 to 2% of a single mouse spleen, or about 50 to 100 microliters of whole blood. And obviously, if you're able to add these analyses into your study, it does enhance the data set. There's a lot of different things that you can use that data for assessing. So here's some example data. This is just looking at splenocytes and trying to characterize just the lymphocyte portion of those splenocytes. So this is forward and side scatter. This forward scatter is indicative of the size of the cell, and side scatter is indicative of the granularity or the roundness of the cell. And you can see already that there's distinct populations of cells. Some of these might be lymphocytes. Maybe up here there might be some granulocytes, some monocytes. But if you just take that whole group of cells and look at 2 markers, CD3 and CD19, you can see 2 very clear populations of CD3 positive T cells and CD19 positive B cells. Because you can use multiple markers on the same cell prep, there's also this graph here, which is CD3 again against NK1.1, which is an NK marker, again, 2 very separate distinct populations. So just looking at these CD3 cells inside this box, you can look at expression of CD4, which is on T helper cells, and expression of CD8, which is on cytotoxic T cells. And you can also look at expression of NK1.1 on just the CD3 positive cells. And I'm not sure why I didn't bother to draw a box around here, but you can see there's a distinct population here. Those guys would be CD3 positive, NK1.1 positive cells, and that's indicative of an NKT cell rather. Then you can go further with the analysis and look at rarer cells. So this is from a study we did where we were looking at the expression of specific integrins on memory T cells. So this is in 2 different lymphoid compartments, the Peyer's patches and the spleen. You can see this by forward and side scatter. They look different because there's a different cell makeup in there. But we're going through CD4 for the T helper cells, and then CD44, CD62, CD45RB rather for differentiating memory and naive cells. And then within that memory compartment, we can look at expression of alpha-4 and beta-7 integrin. You can see that in the Peyer's patches, there's very few cells that are expressing this alpha-4 beta-1 integrin, but in the spleen, there's quite a substantial population. So this gives you an idea of the sort of rarer cells that you can look at if you just go through multiple successive gatings. And also, because we're looking at integrins that's involved in cell migration, then you get an idea of how to assess cell migration studies and whether cells are migrating to specific lymph nodes or not, and whether your drug of interest has any effect on that. So just one more example here. This is a colonic lamina propria prep. So this is about as dirty as cell preps get. There's a lot of dead stuff in this prep. This is a lot of dead cells, cell quants, and epithelial cells, that sort of thing. And that's just because it has to go through a lengthy digestion process to yield a single cell population. But because we're just interested in leukocytes, we can stain for CD45, which is the pan-leukocyte marker, and you get this very nice, distinct population of leukocytes here. And if you just look at expression of CD4 and FOXP3 on those leukocytes, you can see there's a very nice separate population of CD4 events there, and within that CD4 population, there's a few that are FOXP3, and those would be probably Tregs. So from a kind of a horrible-looking cell prep, you can go through some gating and get a really nice readout of a relatively rare cell type. At this point, I just want to talk a little bit about a little example in our oncology studies. I'm not going to cover oncology in great detail because that's for another webinar, but the field of immuno-oncology is very popular right now, and so I just found this little quote from a review, and it's talking about, I'll just highlight the relevant parts, it's talking about how the tumor is able to directly induce functional Tregs to try to suppress anti-tumor immunity, so that if you want to get a good strategy for fighting the tumor, you've got to induce specific anti-tumor immunity, but you've also got to reduce the tumor-induced immune regulation. So there's various ways you can look at that, but I'm going to show you how we looked at Treg influx into the tumor. So again, this is fact analysis. This is a solid tumor digest, CD4 and CD3. So in this double positive gate here, these are T helper cells. And then within that T helper gate, you can look at CD25 and CD39. These are surface markers that are also expressed on Treg cells. So we're not looking at FOXP3 this time. This is a bit of a surface stain version of looking at Tregs. And treatment with the vehicle, you can see that there's a substantial population of Tregs present. The treatment with a checkpoint inhibitor, and that population of Tregs is vastly reduced. So not only can you look at, in your tumor studies, not only can you look at tumor size and survival, but you can also verify the mechanism of action of your drug by doing some very basic facts analysis. So very quickly now I'll talk about cell sorting. Fluorescence activated cell sorting is the same principle as flow cytometry, but instead of just looking at the data on a screen, you actually get the cells back afterwards. So you can draw a gate around the cells that you want to keep, and the software and the machine will be able to isolate those cells within that gate, specifically charge them, and then either attract or repel them from a set of voltage charged plates, and that's how they get into separate collecting tubes. So you can have a tube that has the cells that you're interested in, and a tube that has all the rest of the cells from the prep that you maybe just throw away. Then similar to that is magnetic activated cell sorting. So instead of a fluorescent bead on your antibody, there's a magnetic bead. You cover the cell in antibody, so you're essentially magnetizing the cell. You pass it through a column that is embedded in a magnetic field. All of the labeled cells are retained in the column. So you can have negative selection where unlabeled cells are the ones you want to keep. They drift through and you collect them. Or you can have positive selection where the labeled cells that you are interested in stay in the column. You then remove the column from the magnet and elute them and then collect them that way. So while we're on cell sorting, I just want to highlight a model where we use these two methods of cell sorting, and that is the adoptive transfer colitis model. I'll give some more detail on this model later, but I just want to talk about the cell sorting. This model's been around for a fairly long time. The classic way to sort these naive T cells that are required for the model is to use CD45RB high as your criteria for defining a naive T cell. So here's a spleen prep, again, forward and side scatter. You first of all do a CD4 pre-sort by magnetic separation, and that just enriches the CD4 compartment. So now you've got a population of about 90% CD4 positive cells. You run those through the cell sorter, and you draw a gate around the CD45RB high population. This is CD45 along the x-axis here. And those cells are collected, and those are the naive T cells that you inject into the mouse. When you do this with a cell sorter, you get around 96 to 97% purity. And you can see that the CD45RB expression is sort of a smudge. There's very few truly negative. These ones are positive, but they're just low, and these ones are memory cells. So if you try to do this method with magnetic sorting, you would label all of these cells, and then you'd end up with a population of mixed, naive, and memory cells, and then that would not induce disease. And I'll come on to that later when I talk about the model in more detail. But the method for sorting that we use at biomodels is actually magnetic, but using different markers. So we use a combination of CD62 ligand and CD44, which are two markers differentially expressed on naive and memory T cells. So as you can see in this pre-sort graph here, these naive cells, CD62 ligand high, CD44 negative, and there's some memory cells in there too, CD44 high, CD62 ligand negative. After the sort, most of the cells are in this naive quadrant right here. So exactly the same cells, but looking at them assessing CD45RB expression versus CD44. After the sort, you can see that in the CD45RB high box right here, that's about 97% of that population. So regardless of whether you do the fluorescent cell sorting or the magnetic sorting for this particular cell type, you are getting a cell population at the end of it, which is about 97% CD45RB high cells, and those are naive cells. So just to compare and contrast those two methods, the classic sort involves magnetic and fluorescent sorting, and that makes it slower. It's positive selection, so the cells that you get out at the end are actually tagged by antibody CD4 and CD45RB. And it's a relatively harsh process because of the shearing forces involved in running through a cell sorter, so you get a slightly lower yield and more cell death. One thing I haven't written on here is that with a cell sorter like this, you can get a very, very, very pure population because you can set the size of the gate to whatever you want. Obviously, that's at the expense of yield. The magnetic sort is a one-step process, so it's much faster. The cells you get out at the end are untagged by antibody, which is important because that can affect the expression levels of these markers if you bind antibody to them. It's a relatively gentle process because it's basically just passively dripping through a column, so you get a higher yield and higher viability. And then, like I mentioned, you get the same yield, basically. And another thing that I didn't mention here is that because this step just uses a magnet and it's very small and compact, you can do the whole thing in a biosafety hood, so it can be sterile. And that's not super important for the way we run the model normally, but it could be important for studies with motobiotic mice, and that's something that we are doing more and more of now. Okay, so I'm going to move on to the actual animal models, and I've got an example of 3 of them and how we've used some of these techniques to just enhance them and get more high quality data. So first of all, GVHD. This is graft-versus-host disease. It's a serious complication of patients who've undergone an allogeneic hematopoietic stem cell transplant. About 80% of patients will experience acute GVHD, and of those people, 30 to 70% go on to develop chronic GVHD, and that is the leading cause of death in the acute phase survivors. It is systemic immune activation, so it involves multiple organs, and it is dependent on the presence of antigen-specific T cells in the graft, so it's coming from the donor. Importantly, the host is immunocompromised. That is required for a successful engraftment because if there's a fully intact immune system, you will just very likely reject the graft, which is obviously no good. But also that immunocompromising potentiates donor T cell activation. So there's a fine balance to be struck between effective engraftment and reducing GVHD. So, okay, the model, very briefly, we use BAL-C recipient mice. They get a lethal dose of irradiation, and then we take bone marrow and splenocytes from C57, so these mice are MHC mismatched. We deplete the CD3 cells from the bone marrow and then mix them back in with the splenocytes at a specific ratio and inject a specific amount into the host mice, and then the host mice spontaneously develop GVHD sometime after the injection. And then just to reiterate how this sort of FACS analysis cell sorting is a very simple, very straightforward, very quick process. This is the bone marrow prep pre-depletion and post-depletion, and basically to run this this FACS stain on the cells took about an hour or so, and it just verifies that you have actually depleted the CD3 cells from the bone marrow, and you can go ahead with the injection knowing that your CD3 depletion kit has worked, and you're not just assuming that you're getting the right cells injected. So the GVHD score is a big readout in this model. Obviously, I just want to go over that for a little bit. So it is 5 parameters: weight loss, activity, posture, cell texture, and skin integrity. All of those are scored individually on a scale of 0 to 2, 0 being normal, 1 being moderate, and 2 for severe. And then they're all summed together, so you get a composite score from 0 to 10. Okay, so here's some data, and I'll just talk through this graph quickly, and then I'll go through the GVHD and survival graphs, which all have the same sort of setup. So all of these mice got CD3 depleted bone marrow, but the yellow line at the top, they did not receive any splenocytes, and you can see that they actually don't get a lot of disease. Then the light blue and the dark blue lines, those got 2 different doses of splenocytes, and you can see that the severity of the weight loss is dependent on the amount of splenocytes injected. Then there's 2 positive controls that we use here. One of them is SK506 or tacrolimus, which is the clinically relevant positive control in GVHD. And then also an anti-P40 antibody, which targets the Th1, Th17 response. You'll notice that the positive controls aren't doing anything really for weight loss in that model. By the way, both those positive controls were getting the high dose of splenocytes, so they're comparable with this darker blue line. And then if you look at the GVHD, you get pretty much exactly the same pattern, but obviously reversed. So higher is worse GVHD. Again, if you don't inject the splenocytes, the disease is much more mild. And then if you do inject the splenocytes, you get disease. If you inject more splenocytes, you get worse disease. And the SK506 and the anti-P40 are really not doing a lot in terms of GVHD score. But if we look at survival, here's the SK506 as well as the low dose and no splenocytes group. They're all pretty much up at 100% mortality, apart from this one animal that died the day before the study ended, which is just science, I guess. But anyway, this SK506 is doing a lot to inhibit mortality. And so the reason we're seeing effects on the GVHD score and weight loss is because there are side effects of this drug that include loss of appetite and generally just feeling wretched. So the mice are hunched, so their posture score goes up. They don't want to move, so their activity score goes up. So their weight loss is exacerbated, and their GVHD score is artificially increased. But if you look at survival, it does actually have some effect. Unfortunately, the anti-P40 didn't have a significant effect. You could argue maybe that's reduced mortality, but it's not significant. Okay, so we can also run a variation of this model using human cells as the donor graft. And this is important because it allows you to target human cytokines and cells, which are obviously not present in the fully murine model. So in this model, we use an NSG mouse, which is a severely immunocompromised mouse. They get a very small dose of irradiation, just enough to supply a little bit of tissue damage and inflammation. And then we isolate PBMCs, that's peripheral blood, I should say PBMCs, peripheral blood mononuclear cells from the blood. That includes T cells, B cells, and other antigen-presenting cells. So it's a pretty full complement of cells. And then they get spontaneous GVHD after that. And then just very quickly going over the data, it's very similar to what we see in the fully murine model. In this study, we assessed 3 different donors, green, red, and blue for donors 1, 2, and 3, and then at 2 different doses, high and low, the light and dark lines. So 2 things to see from this graph. Basically, there's a large effect of just the donor that you're using affects the severity of disease, and also the number of cells that you transfer affects the severity of disease. Same pattern again with the GVHD score and the survival. So when I saw this data, my thought was that I would do some FACS analysis on the PBMCs and see exactly what it is we're injecting into these mice, because we're injecting the same total number of cells, but is there a different makeup? Does one contain more T cells, for example? And maybe that can predict the outcome when you're using different donors. And so we did this characterization, and basically there's no correlation. So why does donor 1 induce less severe disease than donors 2 and 3? I can't really see any distinct pattern here that would explain it. One thing I did find out was that donor 1 is actually a guy in his mid-60s, whereas donors 2 and 3 are a man and a woman in their mid-30s. So, maybe it's got nothing to do with the fancy fact analysis. Maybe it's just that they're just coming from different people, basically. So sometimes it doesn't work out. But what we do actually now with our humanized GVHD model is we have a large bank of one particular donor, and they're stored frozen. So if you want to do repeat studies and get consistency over the course of several different studies, you can use the same donor, and that way you get that consistent level of disease induction. Okay, so moving on to the next disease model, EAE, that's experimental autoimmune encephalopathy. That's the mouse model of human multiple sclerosis. It's a chronic autoimmune inflammatory disease of the CNS, and it's characterized by these demyelinating lesions. The majority of human cases are the relapsing, remitting disease course, which can later transform into a chronic progressive disease over time. But a few people present, the first presentation of disease is a progressive form. The etiology is currently unknown, but obviously multifactorial, and genetic susceptibility does play a large role. So the HLA that your cells are expressing, copies of genes involved in T-cell activation and signaling, maturation, that sort of thing. And then it's also known that this disease has a strong Th17 skewed immune response profile. So here's the model. Here's the mouse again. You basically inject a specific peptide along with an adjuvant, CFA in this case, along with pertussis toxin, and the mice develop spontaneous acute progressive disease. If you decide to go with a different peptide, this PLP antigen, again with adjuvant, you can induce a chronic relapsing model, and we run both of these models here at Biomodels. So I'm just going to show some data that covers this model 35 to 55 acute progressive model. So here we've got the blue line is the vehicle-treated mice, so they have disease, but they're not treated with any particular therapy. And then the red line is mice treated with fingolimod, which is also used to great effect in the clinic. This is just weight loss, but you can see that there is a distinct rescue of the weight loss when the mice are treated with fingolimod. We look at the EAE score, which is just a score from 0 to 4 that charts the extent of paralysis. As you can see, these mice that are untreated, they just gradually increase over time, but the mice treated with fingolimod, there's essentially zero disease, which is fairly striking. So if you look at the histology on the spinal cord, this is the vehicle-treated mouse. There's a lot of inflammatory infiltrate that's highlighted by these black arrows. In the fingolimod-treated mouse, there's very little inflammatory infiltrate. This looks very, very clean. And just for comparison, here is spinal cord from a naive mouse. So this mouse did not even receive any disease induction at all. Okay, so this is a little diagram from a Nature review that shows some of the proposed mechanisms of pathogenesis for EAE. And basically up here, a sort of a rogue T cell is being activated by exposure to these myelin-associated antigens and this activated APC here. This T cell becomes activated, and it doesn't realize that it's against autoantigen. It exits the lymph nodes, goes into the bloodstream or into the CNS tissue, becomes reactivated, and recognizes its antigen on the myelin sheath of neurons and starts attacking it, causing these demyelinating lesions that you see. So fingolimod, actually, one of the mechanisms of action is that it inhibits movement of the T cells out of the lymph nodes. So can we verify this mechanism of action by using a simple fact analysis? This is a horrible title that I've got here, but this is central nervous system draining lymph nodes fact analysis. That's what all that stands for. This is the forward side scatter plot again of the lymph node, and then looking at CD3. So these are the T cells in this box right here. Within that gate, you can focus in on the CD4 T cells. These are the T helper cells, and looking at CD44 and CD62 ligand. Again, those memory markers. You can isolate populations of naive cells and then also central and effector memory cells, and then just quantify those across a number of different animals, and you get this data. So looking at just the naive population, obviously in mice that did not receive disease, their lymph nodes have slightly more naive T cells recirculating through them than the mice that have active induced disease going on. Looking at central memory, this population up here on that plot, there's really no difference. I think it's not surprising that animals with active disease after several weeks have more central memory cells, slightly. But then if you look at these effector memory cells, these are the ones that are really doing the damage. The naive mice obviously have a relatively low percentage of these cells because they have no specific immune activation state going on right now. In the vehicle-treated mice that have active EAE, there's a significant amount of effector memory cells in the lymph nodes. But in the fingolimod-treated mice, there is, again, a further significant increase, indicating that those cells are being retained in the lymph node. Obviously, you could go further with this analysis. You could look at the blood, you could look at CNS and see that there's less cells in there in the fingolimod group, and that would indicate that they're not getting out of the lymph node and getting into the tissue or into the circulation. You could do antigen-specific T cells for the MOG 3555 antigens that you injected. So there's a lot further analysis that you could do here. So back to the outline. So the last disease model I'm going to present right now is IBD. So inflammatory bowel disease, a bit of background. It's a whole spectrum of different disorders affecting the gastrointestinal tract, characterized by alternating periods of relapses and remissions with diarrhea, abdominal pain, bleeding. It's clinically diagnosed by endoscopy. And again, with so many of these diseases, the etiology is unknown, but it's obviously affected by the environment and genetics. This just shows a few of them, or one or more, usually multiple factors play into the pathogenesis of these diseases. They can be divided into two subdiseases: ulcerative colitis, which is restricted to the colon, mucosa, and submucosa, and is characterized by this Th2 profile. And then Crohn's disease, which can affect any part of the intestine and indeed extraintestinal tissues such as the esophagus and the stomach. And it's granulomatous in presentation, and it's characterized by this Th1/Th17 cytokine profile. So the models of IBD that we run here are, we run a lot of them. A lot of them are these chemically induced models, so DSS, TMBS, etc. I'm not going to cover those now, but they're covered in at least one of our other webinars. If you would like to go to our website, you can look at those webinars. Right now, I'm going to talk about the adoptive transfer model, which is an induced chronic model. The mice develop disease at about 3 to 5 weeks post-cell transfer, and it replicates many aspects of human disease, including, importantly, small bowel involvement, which isn't really seen in the chemically induced models. It can be treated with clinically relevant therapies such as anti-TNF and antibiotics. And sort of an advantage and a disadvantage of this model is that it really does focus on just T helper cells. So there's no involvement of T cells or CD8 T cells. But also, so that's a bit of a disadvantage, but it's also an advantage because you can really hone in on the effector functions of those T cells and their regulatory functions as well. Okay, so the model itself involves the adoptive transfer of naive T cells into mice that lack T cells, so RAG knockouts or SCID mice. We use a RAG2 knockout as the recipient and a BALX6 as a donor. So it's a syngeneic graft. So there's no GVHD. It's just the naive T cells going in and not being instructed to behave properly, basically. And as I went over before, the naive T cells can be isolated using a variety of different markers. We focus on CD52 ligand, CD44. You really do have to have a very pure cell prep to induce disease. If there's memory cells in there, if there's Tregs in there, then they'll inhibit disease formation. And in the literature, like I said before, this model's been around for a long time. The main readout in life is weight loss, and that's used to confirm T cell engraftment. But also histology is the main endpoint readout, which is obviously a very, very good readout, but you have to wait till the end of the study. So just using those two is not ideal. I'll come back to that in a bit. So here's some data, the weight loss. So I'll go through this graph in a bit of detail. Again, so the yellow line is the sort of naive control. These mice did not receive a cell transfer. They just gain weight steadily throughout the study. Likewise, the red line, mice that received memory T cells get no disease. Then we've got this light blue and the dark blue line, which received 2 different doses of naive T cells. they get either sort of normal or severe disease. And then these two different positive control antibodies against TNF-alpha and anti-P40, they also lose a little bit of weight, but not as much as not quite as much as the untreated disease control. If you look at compare the weight loss over time, there is no significant effect of these antibody treatments, which is quite disappointing. But that's just weight, and as I said before, weight might not be the best readout in this model. So looking at the histology, then this is the disease-free mouse. This all looks very healthy. This is the mouse that got the 500,000 naive T cells. There's a lot of inflammation, a lot of tissue destruction here. If you look at these three slides, this is the memory positive control, and these are the anti-TNF, anti-P40. They look very, very healthy, almost as good as the completely disease-free control. So while the weight loss isn't showing very much difference, certainly the histology is enabling you to see a significant effect of those positive control antibodies. And then when you score it for inflammation, edema, goblet cell depletion, and epithelial damage, and then this is just a sum score, you see in many cases for both of these TNF-alpha and anti-P40, you get a significant reduction from the untreated disease control. And just to remind you, this is what the weight loss assessment looked like. There's basically no significant difference, no real effect at all on the weight loss. So just to remember that, and I'm going to talk about the model as it stands in the historical literature. This is a paper, a review of this model that's been published by this group. We've been working on this model for a very long time, and it's actually the model that we based our model on. They describe 4 different sort of broad disease outcomes in this model. The typical disease, which is about 50% of the mice, that's weight loss between 3 to 5 weeks post-transfer, colonic inflammation, and T cells present in the spleen at the endpoint. Then 20 to 30% of the mice get severe disease, which is a rapid onset of weight loss, and possibly have to euthanize those mice because they're too sick. Then this that I'm calling sort of mild disease, or disease without weight loss. This is the mice look like they're not getting particularly sick, but when you sacrifice them at the end of the study, they have colonic inflammation by histology and then presence of T cells in the spleen. And then this last group here, this small segment here, no disease, so no weight loss, no T cells in the spleen, and no evidence of disease by histology. And I think that's probably just due to failure of engraftment. So this is the situation we get at biomodels, and the good news is that it's very, very similar to what's seen in the literature, which is nice because it means we're running the model correctly. But the problem with this picture is that only 50% of these mice are really getting the disease that we want to see. So I think there's got to be something we can do about this model to make it a bit better. So 3 questions then. Why is weight loss used as a corollary of engraftment? And the answer is that it's very easy to measure, and you can measure it every single day. If an animal doesn't lose weight, you just look at the histology and the splenocyte CD4 count at the endpoint, and if you don't see anything there, you can just retroactively remove that animal from the study. Kind of a waste of an animal and time and reagents, but that's how it goes. What causes the variability in the disease outcome? We know that it's influenced by the number of transferred cells, so my theory is that the difference in disease outcomes is just due to differential T-cell engraftment, and that's based on factors that you can and can't control, so there's not a lot we can really do about that. And then, can this model be enhanced? Yes, absolutely. So when we run this model of biomodels, we run at least one in-life blood draw to assess engraftment, and we usually do that before the onset of any therapy, and I'll come back to why that's important in a bit. We also add in-life serial endoscopies, which gives you it's nothing to do with immunology per se, but it does give you a direct readout of inflammation, as opposed to weight loss, which is an indirect readout, and it's also very clinically relevant, obviously. So focusing on this in-life blood draw, it's a very small amount of blood, so you can do multiple blood draws throughout the course of the study. And we look at 3 different markers, but we're really focusing in on this CD4 because we transferred in naive T helper cells, which is CD4 positive. So mice that received the transfer, you go through this gating, and you can see a very robust population of CD4 positive lymphocytes in the peripheral blood. And just for comparison, naive mice that received no cell transfer, they don't have any CD4 cells in the blood, which is obviously what you'd expect. just to remind you about this, this slide where I showed you these 4 different disease outcomes. So you've got that back in your head. Now looking at that weight loss data again from that earlier slide, just now focusing on these yellow lines, which are mice that did not receive any cell transfer, and the dark blue lines, which are mice which did receive the transfer. And this graph is every single mouse gets its own individual line. So these 2 mice at the top here, they had on day 14 a peripheral blood CD4 count of 0 and no endoscopy score, and I would call that failure of engraftment. This mouse right here didn't really lose a lot of weight, gained weight overall, had a low CD4 count on day 14, a low endoscopy score. I would call that disease without weight loss, or that mild disease. The vast majority of mice in this study had a robust T-cell count on day 14, robust endoscopy score. That's the typical disease. That's what we want to see. And then these 2 mice here, they had a higher T-cell count on day 14 and severe disease. Those 2 mice actually had to be euthanized early. So there is a correlation then between the day 14 CD4 count and then the endpoint disease outcome. At day 14, the correlation is very poor, and I'm just looking at these two ways of sorting the cells just to show you that they are very equivalent. CD45RB high using a flow cytometer, and CD62 ligand CD44 using just magnetic columns. So at day 14, the correlation is obviously very poor, but correlating that day 14 CD4 count with successive days weight loss, the correlation becomes more and more strong as you go through the study. So in this study, we went out to day 63. In this study, we went out to day 49. By the end of the study, there's a very strong correlation with the endpoint weight loss and that day 14 CD4 count. So this suggests that the day 14 count can predict the endpoint disease severity, weight loss, and also endoscopy score. So in conclusion, for the enhancements to this model using just this very, very simple facts analysis, there is this correlation between the early CD4 count and the disease outcome. Usually in this model, we start treatment around about day 14 or later. So we can do this assessment before the onset of treatment. So you don't need to retroactively remove animals from the final study analysis. You just, at day 14, if they don't have a T-cell count, you just get rid of them from the study. So you don't waste mice, you don't waste reagents, you don't waste time. Importantly, you can randomize the mice based on their weight and their T-cell count so that you make sure that each treatment group is as equal as possible with regard to mice that are going to go on to have milder or more severe disease. And that makes each group more comparable with the vehicle group, and so it enhances the power of the study. All of this together obviously just leads to more efficient study design and execution. That's the end of the talking about the individual models. So in summary, for the entire talk, I've shown multiple existing animal models that can be enhanced with immunological techniques, and that's basically these apply to any model where it's advantageous to characterize cells or assess cytokine concentrations, etc. All the things I've gone over today are very well established, very straightforward techniques: flow cytometry, cell sorting, and ELISA. Most of the things we do here are commercially available kits. There's no, it's really sort of very, very simple, very straightforward protocols that you just need to know the immunological background so that you can target the right markers and the right cell types, etc. If you add these assessments into your study, it just adds multiple additional data points. So you can look at in-life and terminal time points. You can predict disease severity. You can confirm the mechanism of action, and you can also, importantly, strengthen the clinical relevance or translatability of your model by showing that your drug of interest is working with the same mechanism of action as it works in the human condition. So with that, I'd just like to thank everyone at Biomodels, the scientists, especially Steve Sonis, who is our CSO and the founder of the company, and Greg, the COO, Ashley and Jessica, who basically keep everything running smoothly and stop me from forgetting stuff, and all our technical staff, which these are the guys that carried out all of the work, basically, and got the data for me for my presentation. So with that, I'd just like to say any additional questions or further information, please feel free to email me, dbill@biomodels.com. And please go visit our website. There's a lot of data on there, a lot more information that cover other models. So thank you very much for listening.
Thank you very much, Dr. Beal. I'd like to thank you, Dr. Beal, for your excellent presentation. I'd like to thank Biomodels for sponsoring today's web symposium. And most of all, I'd like to thank those of you who came and spent some time attending today's webinar. I hope you got some answers to your research questions. Thank you again, and have a great day.